itself is organized. Today's program is organized under his guidance and supervision. So, sir, your invaluable support and guidance is always cherished by all of us. So, thank you very much, sir. We have with us our guest speaker, Dr. Aneesh Thakur. Uh, he is graduate of CSK Himachal Pradesh Agriculture University, and then he did his PG from there only in the Department of Veterinary Microbiology and Immunology. He earned his PhD from Technical University of Denmark, and presently he is working as assistant professor in the Department of Pharmacy at University of Copenhagen. So just after completion of his PhD in 2012, he has a short stint of 3 or 4 years as assistant professor at CSK Himachal Pradesh Agriculture University, and then he joined for postdoctoral assignment at University of Copenhagen. So, as we all know, mankind is undergoing or facing, experiencing a challenge in the form of COVID pandemic, which could be considered as a once in a century kind of problem. So, in India, also it has wreaked havoc, and we have lost many people because of this COVID. uh, Lockdowns are new normal under this condition. It all started, I suppose, in 2019, in November, December from China, and then it spread all over the globe. So it's a very difficult situation, and we all extend our gratitude to the scientific community who worked day and night and developed a vaccine which came as a big respite. So uh, in India also we could come up with two vaccines like Covaccine and Covishield. So initially when vaccine came, uh, the compliance or the say enthusiasm for taking vaccine was very less, but we met with the second wave of COVID, and that again wreaked havoc in our country. Then uh, now we are trying. India is going under massive vaccination drive, but there is a lot of confusion, and these uh, time of social media, uh, a lot of information is coming from different part, and that creates a lot of confusion. People are not very clear that which vaccine to go for, what should be the dose interval, what which titer is going to be protective, or how long this after taking two vaccine one could think that he's protected. What about those who got both the doses of vaccine and even got infected? So a lot of scientific data is coming up. The science related to the COVID is maturing, and uh, to answer all these questions, what better than someone who is related to the development of vaccine, even like a person like Aneesh Thakur, who has his expertise in vaccine design and delivery, especially based on nanoparticles. So under the behest of our director research, we established a communication with him, and I'm thankful to him for agreeing to deliver this talk related to the COVID vaccine. So before we go for presentation, I'd like to invite our director research for an brief opening remark, and then we'll continue with the presentation. Ravindra Kumar sir, thank you.
Thank you, Dr. Pankaj. In fact, we had already chit-chat before the start of the session, and I have apprised Dr. Aneesh about various developments taking place in our university. I would like to just further welcome, first of all, I'd like to welcome Dr. Aneesh Thakur on behalf of Bihar Animal Sciences University for accepting our request to deliver this talk. And of course, our university, as this COVID situation started, took a lead under the dynamic leadership of our Vice Chancellor, Dr. Rameshwar Singh, that under the YouTube channel, we have formed a repository of all e-contents. all the e-contents have been put together at one place, and this is being easily shared, available. All the lectures which are being delivered has been uploaded, so that the students, if they miss some of the lectures, they can visualize later on. And all these lectures organized under NAHP too have been put at one place. So that's a big achievement. We have a very huge number of repository of e-content over there, not only for our faculty, scientific faculty, but for our farmers also. Some of the contents is available on our university website. And under this NAHP program, we have responsibility to develop, take initiatives on the human resource development, which have been appreciated during reviews, annual reviews of the project. So we have taken, of course, that has taken a little bit setback due to this COVID, but still we are organizing such webinars for the benefit of our faculty, and that has helped us in developing national and international linkages. Last year, 3 faculty members from our university visited 2 to US and 1 to New Zealand for 1 month training under this program. So that's and similarly, some of the faculty members were deputed to other universities like CCMB Hyderabad and Covas Palampur, and we are still hopeful if the things normalizes, we'll go ahead further. So I'll not take much of the time. I once again welcome all our faculty colleagues, students, our guest speaker, Dr. Aneesh, and I would once again request Dr. Aneesh that, of course, after the today's lecture, we'd like to have more lectures on the topics specifically on which you are working. Sure. Very shortly, Dr. Pankaj will be in touch with you. So once again, thank you very much. Dr. Pankaj, go ahead, please.
So now I invite Dr. Aneesh for his presentation. Sir, please.
Yes. All right. Can you see my screen?
Yeah.
Yeah, and you hear me well. Yeah. Okay.
Yes.
I would like to thank Professor Dr. Ravinder Kumar, the Director of Research, for the kind invitation to present this lecture today, and also Dr. Pankaj Kumar for the nice introduction, and also for the correspondence we had over the past few days for this lecture. So I think I'm going to give an overview of not just the vaccines, but actually the protective immunity, because that's one of the fundamental questions people have been asking now, that how long these vaccines are effective for. So, and then focusing on the vaccines which are available in India. So if you have any question, please interrupt. I think we can just keep it like a discussion if you want. So I can just stop and answer those questions. Otherwise, we can always discuss the questions towards the end. Okay, so I think I'm going to give an outline here. So I will be talking about a little bit about the vaccines in general, that what are vaccines, how do they work, the different type of vaccines, including the different type of vaccines which have been made against COVID-19, the vaccines which are available in India, and the correlates of protection. And finally, I will sum up with what are my thoughts about natural infection versus the vaccination. So as you know, that vaccines are the proven host defense against the infections. It has been known for centuries that vaccines have contributed a lot to the public health. And this can be judged also from the fact that looking at this graph, this is the incidence of mumps, measles, and rubella in 1980s. You can see the graph over here. And there were many, there were a lot of 4 million deaths in 1980s. But thankfully to the vaccines, now the incidence of MMR has gone down. And it's only a few 100,000 or few million cases every year have been reported because of this infection. So vaccines have contributed a lot to bring down this the disease incidence. Another beautiful example of how the vaccines have contributed to public health is the polio disease. So as you can clearly see that in the 1980s, the incidence of polio was quite high globally. And now towards this century, the incidence of polio has really gone down. And even in India, no cases of polio has been reported since 2014. So which clearly reflects the ability of vaccines to prevent infections. So how vaccines stimulate a protective immune response is that the vaccines, so on the x-axis, y-axis, we have the antibodies in the T cells, and on the x-axis, we have the timeline. So what happens during an initial infection due to a virus or bacteria is that our immune system responds to the first infection. And then we, after like a few days, then we generate an immune response, which is characterized by either antibodies or T cells. And these antibodies are T cells which they generate in about 2 to 3 weeks time. And then they induce the protective immune responses, and then that's the levels of these antibodies and T cells wane down, they goes down, and then you have the level of antibodies and T cells which are protective against the ongoing infection. And in months later or years later, if you get the same virus or same bacteria or the same infection, your immune system will recognize it and then again generate a very strong immune response. It does also take some time, but it's rather quicker than the first infection. And what we call it as the immunological memory, that the immune system has the ability to remember the infection. And this immunological memory is the basis of all the vaccines which have been developed so far. So going back to the history of vaccines, so if we have to consider that how effective are the vaccines, we have to go back to the human evolution. So human evolution started about 3 million years ago. And then since that time, the average life expectancy of a human being was less than 30 years, roughly around 25 years. And by the time the humans started to undergo evolution and came out of Africa and nearly around 100,000, it was still the same. So when the Homo sapiens, they undergo evolution and came out, the average life expectancy was still 25 years. However, something dramatic happened during the year 1796 in the United Kingdom, in which the physician Edward Jenner, he did the first form of vaccination. So what he did, was that he observed that the people actually the milkmaids who work with the cowpox, work with the animals, the cows, they develop a mild form of smallpox, which is called the cowpox. And he realized that this cowpox is kind of protects these females, these women from cowpox, the real the pox virus infections. So what he did is that in 1796, he deliberated, infected a boy, James Phipps, with the pustules or the pus which he collected from the cowpox lesions. And then he injected into this boy. And then after about 2 weeks time, he then deliberated this boy with the smallpox lesions. And what he observed that this boy was not, did not contract the smallpox disease, and this form of vaccination actually prevented the boy from the smallpox lesions disease itself. So that serves as the basis of first form of vaccination. So since then, the average life expectancy since the introduction of vaccines have increased to about 80 to 85. Even in India, the average life expectancy of the average Indian is about 70 years. So, of course, there are many factors beyond the vaccines which have contributed to this, but vaccines have contributed the most, or it has been the major contribution, because of which the life expectancy has increased to 80 or 85 now. And thankfully, because of the successful introduction of vaccines, smallpox was eradicated or completely eradicated from the planet Earth in 1980. And then it's a human disease. Similarly, rinderpest was also eradicated. That's the only veterinary disease which have been eradicated, and again because of the successful vaccines. So coming to the COVID-19, so as you see that, as Dr. Pankaj has already mentioned, that within this time frame, since the virus was first reported in 2019, within a year time, many candidate vaccines have been tested already, and about 184 candidates are right now in the preclinical phase, while 108 candidates have been already in the clinical phase testings in different phases. So these are the number of different candidates which are in the different phases of clinical trials. And about 8 of these vaccines have been approved by WHO to be used under the emergency circumstances as of now. And here is a list of some of the most promising vaccine candidates against COVID-19. And I'm not going to focus on it all, since I would like to focus only on the vaccines which are available in India. That is the Bharat Biotech vaccine, the vaccine from Russia, the Sputnik 5, the AstraZeneca Oxford vaccine, and the recently approved the Moderna vaccine, which is mRNA-based vaccine. So I think I'm going to focus on only these 4 vaccines for today's topic, today's lecture. So before coming to these vaccines, it's important to understand which kind of immune responses are generated when we get virus infections. So typically for any disease or any viral infections, whenever we get a viral exposure, our immune system, especially the innate immune system, responds first. And this response happens within minutes to hours to days of the first exposure to the virus. And this innate immune response detects the virus and it communicates with other cells, and it goes down after some time, after the matter of days and weeks. And by the time the virus is also, of course, there's a kind of competition going on between the virus and the immune system. So if you develop or the immune system is not able to thwart the infection, the load will increase. And after some time, after a few weeks, almost about 7 to 10 days, our adaptive immune system gets activated, which is made up of B cells and T cells. And as the adaptive immune system comes into action, the viral load correspondingly goes down. So that takes about several days from the first exposure until the adaptive immune system activates, and then the viral load goes down. So what happens in the innate immune system is that when any pathogen, for example, in this case, SARS-CoV-2 virus enters the immune cells or the lung cells for that matter, it is detected by the different kind of sensors which are present on the immune cells, which are called the pattern recognition receptors. So once the virus is taken up by these or detected by these sensors will activate downstream signaling pathways within the cells, and then these cells will in turn produce some protein molecules called cytokines and interferons. So these cytokines and interferons in turn have different functions. They will either, one of the function is that these cytokines and interferons will tell other neighboring cells to put a barrier against the virus, so as to prevent the transfer or the multiplication of the virus. These cytokines and interferons can also signal other cells which have been infected with the virus to die. Or one of the major functions of these cytokines and interferons is to recruit other immune cells, especially the immune cells which play an important role in the adaptive immune response, that is the B cells and the T cells. So all of this is happening within the first few hours to first few days of infection. So at that time also the virus was increasing, and an adaptive immune system takes a while to start. So these cells, the immune cells which have been receive the signal from the cytokines interferon, they will pass the information to the special kind of cells which are called the antigen presenting cells. The most important are the dendritic cells, and these activated dendritic cells will then these cells will become activated, and then they will go to the local lymph nodes. In the local or the draining lymph nodes, these cells will in turn activate the CD4 T cells, which are called the helper T cells. And these helper T cells will start what we call as the cell-mediated immune response. That is, these helper T cells will in turn activate a special category of T cells, which are called the CD8 T cells or cytotoxic T cells. And the function of these cytotoxic T cells is to kill the virus cells or the virus infected cells. On the other hand, these helper T cells can also activate the B cells, which is called the antibody-based immunity. The B cells, the function of the B cell is to produce the antibodies through plasma cells. And these antibodies will in turn neutralize the virus, and that's why the so-called neutralizing antibodies. So this constitutes the adaptive immune response to virus infection or to any pathogen in this case. And it takes, as I said, it takes some days. It takes about 7 to 14 days minimum for these things to happen. And that's why in this period of time, 7 to 14 days, you still have increased virus load in your maybe in your nasal cavity, in the lungs, or maybe in the circulation, depending on whether it's a mild disease or acute disease. So how a vaccine generate an immune response against COVID-19 or any other virus infection? So vaccines essentially mimics what happens in a natural infection, but in a safe manner. So whenever, as soon as we get a vaccine into our muscles, the vaccine antigen is released into the muscle cells, and it's taken by the antigen presenting cells, especially the dendritic cells, which are localized in the muscle tissue. These muscles, these dendritic cells, which have taken up by the pathogen virus antigen, they will become activated, and they will go to the nearest lymph nodes. In the nearest lymph nodes, they will in turn activate the CD4 T cells. As I mentioned earlier, they are the helper T cells. Their function is to help other cells. So these CD4 T helper T cells will activate. They will help the CD8 T cells to become activated. Once activated, the CD8 T cells will transform into the cytotoxic T cells, which will kill the viruses. Or a population of these these CD8 T cells will remain in the body, which then they will form the memory T cells. As I mentioned, the CD4 T cells will also help another category of cells, which are called the B cells. B cells in turn, they will proliferate and multiply in number. They will become the plasma cells, which produce the antibodies. And these antibodies, they will circulate in the blood, called neutralizing antibodies. However, a fraction of these antibodies will also go to the bone marrow and stay there, which will form the long-lived plasma cells, or these plasma cells will serve as the continuous supply of neutralizing antibodies for months or years post-infections. So essentially, vaccines mimic what is happening in the natural infection, but they mimic this thing in a very safe manner. So, but how vaccines actually, so this is the vaccines I explained that they work at the individual level, but how vaccines actually work in the real world is explained by this figure, in which so this group population is all the people who have been not vaccinated. And if we have one infected individual, which is marked in red, so this infected individual can spread the disease to all the other individuals in this herd, in this community. And this is what you see here. This is what's happened with the SARS-CoV-2 in 2019, that the infection started from one place and then it spread through the rest of the world. However, if we can somehow vaccinate this population against the SARS-CoV-2 and any other virus infection, so this is the second scenario, that if the vaccination is below the herd protection, which is called the herd immunity or the community immunity, if we are immunizing the individuals below the threshold, only few individuals will be, let's say, only half of the individuals will be blue in color, which are still susceptible, and yellow in color, which have been vaccinated. So in this scenario, if you have one infected individual, which is marked in red, this infected individual can still spread the disease to another individuals, which are the susceptible ones marked in blue. It is because the vaccination is below the threshold, the level, which is often referred to as the threshold for the herd immunity. And then the third in a scenario is that when the vaccine coverage is above the threshold of what is recommended for the herd immunity. And if that's the case, even if we have infected individuals in this herd, this infected individual may not be able to transmit the disease because of the herd immunity. Now, the maximum individuals in a herd are immune to this disease, and they will not be able to transmit the disease to other individuals, as seen in this graph, that the infected individual is not able to transmit the disease from the vaccinated individuals. So this is the concept of herd immunity, and the vaccines work in this fashion in the real world. However, it is important to consider what is the threshold for the herd immunity, and it differs from disease to disease. For many of the different viral infections like measles, it's 92 to 95%, which is quite high. That means if 92 to 95% of individuals in a herd should be vaccinated in order to achieve the herd immunity. For pertussis, it's about more than 90%. For rubella, it's again 83 to 85%. For SARS-CoV-1, it was 50 to 80%, and now it is known that for SARS-CoV-2, this herd immunity threshold is average is around 70%. So that means we need to have about more than 70% of the population vaccinated in order to achieve the herd immunity. Coming to the COVID-19 vaccine. So what's in a COVID-19 vaccine? So what are the components, or what exactly is in a COVID-19 vaccine? So broadly, these COVID-19 vaccines can be categorized into three categories. One is the vaccine type called the whole cell vaccines. Whole cell means we are working with the full pathogen, like SARS-CoV-2 virus in this case. Another category of vaccines are the subunit vaccines, that instead of the whole virus or the whole pathogen, we work with fractions of the pathogen, either proteins or nucleic acids or polysaccharides or some peptides, which are immunogenic. And third category of the vaccines are in which we use the genetic material of the pathogen as a vaccine candidate, either DNA or the RNA. So fortunately for the current platforms which are available for COVID-19 vaccines, all of these different platforms have been used, and then many of these vaccines which are available at the moment belong to one of these categories. So looking at the whole cell vaccines, whole cell, as I mentioned, that we start with a full pathogen, and then we try to inactivate this pathogen in the cell lab, in the cells using some kind of chemicals like formalin or beta propionolactone. And the net result is that we have inactivated virus, which is often has to be injected with adjuvant. So examples of these vaccines is the vaccine which is available in India called Covaccine, which is built by Bharat Biotech, which is an inactivated vaccine, which is based on this platform. So another example of the subunit vaccines is a protein-based vaccines in which of course, most of these COVID-19 vaccines are using the spike protein as the target. So what we do in this kind of vaccines is that we identify the target of interest, that is the spike protein antigen, the DNA, and then we insert into plasmid. We recombinantly produce the proteins, and then we inject the proteins with some kind of adjuvant. In this case, it was the matrix M1 adjuvant, and then we generate an immune response. So example of this vaccine is a Nova vaccines, which is awaiting approval to be used in India. Another category of the subunit vaccines are the viral vector vaccines, in which, of course, we still use the spike antigen, but instead of using them as proteins, we introduce this spike protein antigen into a adenovirus vector-based plasmid, and then we recombinantly produce grow them in the lab, and then we have the gene of interest inside the viral vector. And the important feature about this viral vector is these are non-replicating based viral vectors. So that means these viral vectors, they are just used as a delivery systems. They will not cause infection or disease in the host, and they will only deliver the gene or the antigen of interest. An example of these vaccines which are being used in India are the AstraZeneca vaccine, that is the Covishield, and the Sputnik vaccine, the vaccine from Russia. The Johnson & Johnson vaccine is also based on this principle. Now, the third category of vaccines, as I said, are the nucleic acid-based vaccines, in which we can use either DNA, which is introduced into a plasmid, and in this case, it's a spike protein encoding double-stranded DNA, which is inserted into the plasmid, and then you have this the DNA-based vaccine. So, example of this vaccine is Inovio, which is built in this kind of vaccine platform. And then the messenger RNA vaccine is the last category of this nucleic acid based vaccine, in which instead of DNA, we use the messenger RNA, and the spike encoding messenger RNA is inserted into some kind of lipid nanoparticles, and then resulting is that you have the lipid nanoparticles which have the spike protein including messenger RNA encapsulated inside the lipid nanoparticles. And both Pfizer vaccines, Pfizer-BioNTech and the Moderna vaccines are examples of this kind of vaccines. And Moderna has been approved recently, a few weeks ago, to be used in India. So it's very interesting actually. As far as COVID-19 vaccination in India is concerned, this is data from yesterday, and then we have reached around 400 million doses, total doses, and marked in the yellow graphs, and in the red, marked in the red. In the yellow, we have the people who have been vaccinated, who have received one dose of the vaccine, which is roughly about 300 million. And the green, the line show the people who have been fully vaccinated. That means they have received 2 doses of vaccines. However, although we have vaccinated many people, but India, if you look at, only 5.9% of the total population has been fully vaccinated. That means given 2 doses of vaccine. And just 17% of the population has received 1 dose of the vaccines as yet. And then you can compare the other countries, we are lagging far behind in order to achieve the herd immunity. As I mentioned, 3 vaccines have been used in India: the Covishield vaccine from AstraZeneca, the Covaccine from Bihar Biotech, and the Sputnik vaccine from Russia. If you look at the distribution of these vaccines, so majority of the people in India have received the Covishield vaccines, roughly about 338 million, followed by Covaccine, about 47 million people have received this vaccine, and Sputnik have just been introduced, so only few people have been vaccinated with this vaccine at the moment in India. And as I mentioned earlier, that Moderna has recently received approval, and it will be launched very soon from Cipla. And it's a mRNA-based vaccine from Moderna and based in the US. Other candidates which are awaiting approval for being used in Indian population is this Lycov-D vaccine from Zydus Cadila. It's a DNA-based vaccine. And 2 subunit-based vaccine, subunit protein-based vaccines, one from Biological Media Limited and one from Novavax. They will be hopefully approved and being used in the coming days in India. So coming to the first vaccine, that is the inactivated vaccine, the Covaccine, which has been used in India, which is also called BBV152. This vaccine has been developed in collaboration with between Bharat Biotech and National Institute of Virology. As I mentioned, it's an inactivated vaccine. So inactivation means this. There was this strain which National Institute of Virology cultured in the labs from patients, and then they inactivated this strain of virus using beta propionolactone. And what they did in Bharat Biotech is that they introduced this adjuvant called L-gel IMDG. This is the property adjuvant. This adjuvant has 2 components. One is alum, that is aluminum hydroxide, which is one of the most commonly used adjuvant in human in the veterinary medicine. And to this alum, they added a molecule. It's a tricyclic compound called imadazoquinoline. It's a TLR7/8 agonist. And then this vaccine was given to the patient, to the population in a dose of 6 microgram intramuscularly twice, and at an interval of 28 days. So this, as I mentioned, it's a TLR7/8 agonist. So this molecule, this tricyclic molecule, is recognized by receptors, as I mentioned earlier, different kind of receptors which are present in the immune cells. In this case, it is recognized by receptors which are present inside the endosomal cells, the TLR7, the activate, in this case, they activate the other immune cells, and which will generate the adaptive immune response. So the end result is that this vaccine is able to induce very strong antibodies as well as the Th1 or the helper T cell responses. The first data which came out from Covaccine is from a phase 1, phase 2 study in which this vaccine showed that day 0 before immunization and then 28, 42, and 56 days post immunization. So the initial, it was testing 3 microgram and the 6 microgram doses, but finally for the phase 3 testing and what is being used is 6 microgram dose. And as you can clearly see that compared to the basal immune levels before the immunizations, all of these, both the 2 doses, 3 and the 6 microgram, induce very strong the neutralizing antibody levels in the phase 1 and the phase 2 clinical testing. And this was then compared with the neutralizing antibody titers in patients who have recovered from COVID-19, that is the convalescent serum samples. And this vaccine in these 2 doses induced very high levels of neutralizing antibody titers as the convalescent serum samples. So later on in phase 2 testings of this vaccine, Covaccine, the cell-mediated immune responses, that is the T cells, were also measured. And this graph shows that the 3 micro and the 6 microgram doses induces very strong the T cell responses, which produce either the TNF-alpha or the interferon gamma or the different combinations, which are often referred to as the polyfunctional T cells because they produce a different range of cytokines and the chemokines. So that was very encouraging data which came out of Covaccine vaccine. And ultimately, this vaccine Covaccine, this data just came out a few days ago, and then it's still under review. It is under bioarchive, meta-archive. And when this vaccine was tested in the phase 3 clinical trials, it was found that the final vaccine efficacy was 77.8%, which is very encouraging data. So this is the placebo responses, that is the viral loads in the people who did not receive the vaccine. So compared to those people who did not receive the vaccine, the vaccine vaccinated individuals, they were protective. So overall efficacy of this vaccine is 77.8%. So coming to the second vaccine, which is extensively used in India, is the Covaccine vaccine, often referred to as the AstraZeneca and the Oxford, University of Oxford vaccine. And this vaccine is again given intramuscularly, and it has different time intervals from 8 to 12 weeks intervals, and also 2 doses. So as I mentioned previously, this vaccine is based on the adenovirus vector-based principle, the viral vector principle. So what they did is that they introduced the spike protein encoding this genetic material into a viral vector, which is used as a delivery system. In this case, it was a chimpanzee adenovirus vector. It is a virus adenovirus which infects the chimpanzee, and which has been which does not has the ability to replicate. So it is replication deficient virus. So this virus is essentially used as a vehicle to deliver the SARS-CoV-2 genetic material, that is the DNA of the genetic of the SARS-CoV-2 antigenic protein. So how this vaccine works is that once this adenovirus vector is introduced, the adenovirus vectors are usually recognized by the TLR9 receptors in the cells. And once activated, these cells will in turn release the interferons and the chemokines, and then they will activate the helper T cells, which will either activate the B cells, which will produce antibodies, or these T cells can differentiate or they activate the CD8 T cells, which can kill the ongoing virus infection. So the data which came out from the phase 1, the phase 2 clinical testing of this vaccine clearly showed that the vaccine used very strong neutralizing antibody titers, and compared to the convalescent serum samples from the recovered patients, 28 days post immunization. And later on, when the T cell responses were measured, and these are the T cell responses after the first dose, 14 days after the first immunization, and you can see that. So these are the responses which are measured in the peripheral blood T cell responses. So this vaccine has the ability to induce both neutralizing antibody titers as well as the T cells. So very similar to Covaccine, the Covishield vaccines also induce very strong T cells and antibodies. And ultimately, the phase 3 clinical data of the vaccine showed that the vaccine compared to the placebo control is very effective, and the overall effectiveness of the vaccine was 70.4%. And this vaccine was tested in 2 different doses, standard dose, and then each one they tested the vaccine in low doses followed by a standard dose, and then they extended the time interval, and they found that the vaccine efficacy really goes high and high as you increase the time interval between the 2 doses. So coming to the third vaccine, which is being used very recently, is the Sputnik vaccine from Russia, from a Gamaleya Institute or Microbiology and Patna Institute in Russia. It's based on, again, adenovirus vector-based vaccines, but compared to the AstraZeneca or the Covishield vaccine, this vaccine is different in a sense that in AstraZeneca or the Covishield vaccine, both the doses, the first dose and the second dose is given with the same vector, adenovirus vector 5. But in this case, the first immunization is given with the adenovirus vector 26, and the second immunization given with the vector called adenovirus vector 5. And so one of the advantages of this vaccine compared to the Covishield vaccine is that in Covishield, your immune system can recognize the adenovirus 26 in the second dose and generate an immune response. So that can reduce the efficacy of the vaccine, the overall efficacy of the vaccine. But if you use 2 different vectors which can be used to deliver the genetic material of SARS-CoV-2 antigen, that eliminates that possibility. So that means you will not be able to, or your immune system will not be able to generate the immune responses to adenovirus 26 in the second dose, because the second dose is with a different vector. And that result is that you will generate a very strong immune responses, and that's what is Sputnik manufacturer reported. So what happens is that once you are given the first dose, you will the vehicle will release the genetic material. It will undergo translation, and the proteins are produced. You will generate the antibodies, and then in the second dose, you will generate the antibodies which are specific to SARS-CoV-2, but your immune system will not recognize the adenovirus 26 anymore, because now you have a second vector. And this vaccine, when tested in the clinical trials phase 1 and the 2, it was found to generate antibody, neutralizing antibody titers in all the different people in different age groups. And compared to the placebo, you can clearly see very strong immune responses in both males and the females. And similarly, they found that it does induce strong T-cell mediated immune responses. That is, the T-cells which produce the interferon gamma after just 1 dose of immunization. And the overall efficacy of this vaccine was reported in the phase 3 clinical trials, which was way higher than the Covishield vaccine, 91.6%. And it's probably due to the strategy of using 2 different virus vectors to deliver the antigen. Now, the fourth vaccine I'm going to talk about today is the mRNA-based vaccine, which is mRNA 2073, which is Moderna, it's a Moderna vaccine. In India, it would be sold in the name of Spikevax, and it has recently been approved to be used. And this is a mRNA-based vaccine. So instead of using DNA or the proteins as the vaccines, this approach we use is a messenger RNA. Since the messenger RNA is used as a candidate here, it needs to be delivered inside the host cells, and that's why they have used lipid nanoparticles. So these are all lipids which encapsulate the messenger RNA, which is marked in green here. So once delivered into the host cells, this is how these lipid nanoparticles look like. So what happens inside the cells is that messenger RNA which is released from these lipid nanoparticles. It is taken up by the ribosomes, and then the ribosomes are the factories to produce the proteins in host cells, and then the net result is that you have spike protein antigens produced inside the host cells. So these peptides or the proteins are produced. They will taken up by the MHC class 1 or the class 2 molecules, and then they will be in turn activating either CD4 T cells or the CD8 T cells. And as I said, CD4 T cells help in generating the antibodies, while CD8 T cells will have the cytotoxic function to to kill the host cell which has been infected with the viruses. So when this vaccine was initially developed, it was tested in 3 different doses, 2500 and the 250 microgram. And for the phase 3 clinical trials, and what is being used currently in humans is the 100 microgram dose. And you can clearly see that the 100 microgram dose of this vaccine induces neutralizing antibody titers already 36 days post-immunization. Its vaccine was also found to induce CD8 T cells 43 days after the immunizations, that is after the 2 doses of immunizations. And the overall efficacy of this vaccine in the clinical trials, phase 3 clinical trials, was 94.1%. So it's quite high efficacy of this vaccine. So all in all these 4 candidates are really promising and which are being used in India at the moment. But the question is how the vaccines work in real life in general population. So all the data I have shown is from the clinical trials, and we know that in the clinical trials, all these vaccines are very efficacious. The efficacy ranges from 70 to 95%. However, since we have to use the vaccine in the general populations, we need to know what is the efficacy of that vaccine in the real population, the real world, and then it's called the vaccine effectiveness. So vaccine effectiveness differs from vaccine efficacy. So vaccine effectiveness is the actual ability to protect the vaccine in the real world population. So here is a summary of some of these vaccines, and this data is only available for some of the vaccines which have been used. I would like you to focus on only these values, that the Pfizer and the BioNTech and the Moderna vaccines, which are the mRNA vaccines, depending upon what is the readout, whether it's the severe disease or hospitalization or symptomatic or asymptomatic conditions, all of these vaccines have very high efficaciousness, which ranges from about 70 to 90%. While AstraZeneca, when the readout is hospitalizations, so it was 94% effective. This is the data which came from Scotland. Another study done in India and also found that the AstraZeneca vaccine against the symptomatic COVID-19 has efficacy, or the efficaciousness of this vaccine is more than 80%. And for Pfizer and the Moderna studies which were done in US and Qatar, they found that the vaccines have overall effectiveness more than 80% in general populations. So there is no data from which is available from Covaccine and the Sputnik at the moment, but hopefully this data will come out as we progress in the coming days or months from now. So overall, the data show that not only the vaccines are effective in the clinical trials, they are effective, or they have efficacy in the real world setting also. And then if the readout is hospitalization or symptomatic COVID-19, majority of these vaccine candidates, they induce very strong, they have very strong immune responses, and they have very effective, more than 50 to 90% efficacy. So another question is how long this vaccine immunity lasts. So we know that the immune responses, or the immunity against COVID-19 vaccine, COVID-19 infection, it stays for about a few months, 7-8 months, but afterwards it goes down. But what about the vaccine? So unfortunately, not much data is available for this durability of immune responses against all the candidates. However, the data which is available is from mRNA vaccine, which is from Moderna, and it has been observed that the immune responses which has been induced by this vaccine lasts for about 7 months. And this study is also shows that these are, of course, the neutralizing antibody titers, and they found that these titers remain, or these neutralizing antibodies stay in the blood of the vaccinated individuals for about 7 months. So there is another data which is recently came out a few days ago. It's from the Pfizer BioNTech vaccine candidate, and what they observed is that this vaccine is they observed that they got the lymph node biopsies from the vaccinated individuals, and this is the images from the lymph nodes of the 2 other individuals, and they found that the germination memory cells, they reported in these individuals, and they sustained for about 4 months. So they're based on these studies, it is now known that both, at least the mRNA vaccines, they induce very strong immune responses, which are durable and which generates memory in the lymph nodes of the vaccinated individuals. But the data from other vaccines is not available at the moment. Covaccine, we don't know actually how long this immunity will last, but hopefully in the few months' time, this data will come out. As far as the Sputnik vaccines, no data is available. The Janssen vaccine, which is also from Johnson & Johnson, it's from US, they recently reported that they're neutralizing antibody titers remain for about 8 months at the moment. But they are following the studies for the next few months. So now the other question is that, what is the correlative protection of these vaccines? Correlative protection means any immunological parameter or any immunological reading which can tell you that, okay, if you have the antibody titers above this level, then you have protective against the COVID-19. Or if you have the antibody titers below this level, then you are not protected. And again, it's not clear yet that what are the exact or the right correlates of protection against COVID-19 induced immune responses against following the different vaccines. However, some of the studies which recently came out correlated that neutralizing antibody titers definitely have a clear correlation with the protective efficacy of the vaccines. So different vaccines, which they all correlated with neutralizing antibody titers. However, it's not clear yet that what is the exact threshold, what is the exact titer of these antibodies. So hopefully, again, this data will come out. So all of these vaccines, especially the vaccines which are being used in India, like the the COVID-19 vaccine, the Covaccine, and the mRNA-based vaccine, they all correlated with neutralizing antibody titers. So this is very encouraging. So also a similar study also reported that the neutralizing antibody titers, what they observed in clinical trials, they correlated with the vaccine efficacy, and again a linear correlation was observed between the neutralizing antibody titers and the protective efficacy of the vaccine. So this is very encouraging because some of these vaccines are being used in India, like the Moderna, the Sputnik vaccine, and the AstraZeneca vaccines. However, as I said, we don't know the exact titers of the neutralizing titers of the antibodies at the moment. We do know that in monkeys, in macaques, it is 1:250, but for humans, it is not clear at the moment. So now another question is that whether these vaccines protect against the new variants, especially the Delta variant, which is prevalent everywhere now. And then here's a summary of the current the data which is available from the clinical trials at the moment. So if the readout is symptomatic infections, the BioNTech or the Pfizer vaccine has about 79 to 88% effectiveness against the Delta variant. And if the readout is death or hospitalization, then this vaccine is 96% effective. Similarly, Moderna vaccine is 72% against symptomatic infections and 96% against the deaths and hospitalizations. And AstraZeneca is, and the AstraZeneca vaccine is 66 to 67% effective, while hospitalizing that it protects 92%. And the only data which is available from the Bihar Biotech is against the symptomatic infections. So the overall efficacy of the Bihar Biotech vaccine Covaccine is 65% against the Delta variant. So this data clearly shows that all of these different vaccines, they have effect against the Delta variant. And I would like to focus here that this data is based on 2 immunizations, not just the 1 immunization. So it's important to get the 2 vaccines in order to achieve this level of protection against the symptomatic infection or the hospitalization and deaths. So I think this is my last slide. So I would like to just sum up by saying this, that there are differences in the natural infection of SARS-CoV-2 versus the vaccination-induced COVID-19 immunity. So if you have a natural infection, you will definitely get sick with COVID-19, and then it can be dangerous. However, if you have the vaccines, so definitely vaccines reduces the chances of getting a COVID-19 infection. So natural infections, one person can develop the long-term complications, especially the long COVID. However, vaccinations against COVID-19 is very safe in the control immune responses, and then you will never contract COVID-19 infection because of the vaccines. In natural infections, you can spread the virus to others, but COVID-19 vaccination reduces the virus spread to the general population and helps in achieving the herd immunity. So natural infection induces very variable immune responses. So some people can induce very good responses, others can induce very poor immune responses. Vaccination does induce also very variable immune responses, but the vaccine responses tend to be very strong and consistent among the people, among the population. And even the booster immunizations strongly from the natural infections, vaccine tend to increase that immune responses. And then immune system may not be able to recognize the different variants if they have actually infected. However, the vaccinations, since vaccination induce very high levels of titers of neutralizing antibodies, they are likely to cross-react with different variants. So it's likely that if you have been vaccinated, you will be effective against the new variants, Delta variant or other variants. So overall, to conclude, I would like to say that all the COVID-19 vaccines which have been developed and which are being used in India, they're safe and effective. They are not only effective against the original variant, but also effective against new variants like the Delta. So it's really important to get the vaccinations. Even if you are infected, it's good to have the vaccine because vaccines are known to enhance the immune responses which you generate post infections. So it's really important to get the vaccine even if somebody is infected. It's important to maintain the social distances because until we achieve the herd immunity, until we have the 70.8% population vaccinated, we have to keep the distance. So it's important to keep the social distances while the vaccination is being rolled out. And it's important to get the vaccines as soon as possible because it's really important to achieve the herd immunity this threshold of immunity as soon as possible. And then finally, I would like Dr. Pankaj send me a few questions, and then he asked some questions sent me through email, and I would like to answer some of these questions before we take another questions if you have. So another question is that which vaccine to go for and why? So I think based on this data, you are convinced, hopefully you are convinced that all of these vaccines are very good, they are protective. So I cannot point out which vaccine to choose. If you have, it's really important to get the vaccine as soon as possible. So it's immaterial which vaccine, but any vaccine which you can get hold of, it's important to get that vaccine as soon as possible or whenever your turn comes. So what should be the protective titer? I think I have answered that question. So we don't know at the moment what is the exact protective titer of the vaccine inducing of any of these vaccines, but hopefully in a few months' time, we will reanalyzing the data from phase 3 clinical trials. So hopefully we'll get to know the protective titers. And I would like to mention here that protective titers would differ from vaccine to vaccine because all of these vaccines have been developed differently. They have been evaluated differently. Even the method of evaluation of neutralizing antibody titers in these clinical trials were different. So we might see that these titers will be different from vaccine to vaccine. So how long the protective titer will remain? So at the moment, we know that at least for messenger RNA-based vaccines, where the immunity remains about 8 to 9 months. And these studies are still ongoing. So it has been proposed that since the germinal centers are formed in the lymph nodes, they have been also reported in the bone marrow. So it is expected that at least for messenger RNA-based vaccines, these titers or these neutralizing antibody titers will remain for many months or years actually. So what should be the dose interval? So for most of these vaccines which are used in India at the moment, the dose interval should be around 3 to 4 weeks. However, for AstraZeneca, that is the Covishield vaccine, it has been observed that if we extend the dose interval time from convention 3 to 4 weeks to 8 to 12 weeks, the efficacy of this vaccine improves. So you just have to follow the guidelines, and then the interval for Covishield is a little bit longer than other vaccines. So what is the risk of getting COVID in immunized individuals? So it's a very good question. So answer is that vaccines help preventing the transmission of the disease, but you can still have the infection while you have been vaccinated. As I mentioned, it takes about 7 to 14 days, that is 1 to 2 weeks interval before, once you are fully vaccinated, in order to your immune cells to become activated, in order to adaptive immune response to kick in. So for that duration, 1 to 2 weeks interval, you still get, there's still some chance that you can get the disease. And not all the vaccines are 100% effective. So during this duration, 3 weeks, 2 weeks interval after your second dose, you still have a chance of getting the COVID-19 infection. But since I said vaccines are effective, you will not be able to contract the severe disease. It will reduce the symptomatic disease and reduce the chances of hospitalization and deaths. So whether these vaccines are effective against the emerging variants, yes, the vaccines have been shown to work against all the different variants, alpha, beta, and even the delta. And especially for delta variants, vaccines have been have this efficacy from 65 to 80% after 2 doses of immunization. So it was found that if you have only 1 dose, the efficacy goes down. So it's important to get the 2 doses or the full vaccination schedule against emerging variants. So another question I was asked that why mortality is reported in doctors despite receiving both the doses. So there could be several reasons for this. So as I mentioned, so after second dose of vaccines, you are still susceptible for about 2 to 3 weeks until your immune system generates the neutralizing titers and T cells at that particular level. So maybe these doctors, maybe they were within these 2 to 3 weeks interval. The second reason could be that I haven't looked at what the data is that whether they were symptomatic in this period or not. So as I said earlier, that not all the vaccines are 100% effective. That means majority of the vaccines, and this COVID-19 vaccines, they are 70 to 94% effective. And breakthrough infections, there is a term called breakthrough infections. That is all those people which have been vaccinated, but they will still not respond well to the vaccine. So there's chances that anything between 5 to 35% in this case, they will respond poorly, and those are called the breakthrough infections. So probably those doctors they had this breakthrough infection post immunization, and then they have mortality because of this reason. And then the other question was that emergence of new variants like Delta and the Delta Plus. So I haven't seen any data on the Delta Plus, but it seems that it's this new vaccines or the vaccines are going to work against this strain. We just have to maintain the social distancing plus the general hygiene. But this variant is just more transmissible. But other than that, there is no data at the moment that the vaccines. And I don't see any easy reason that vaccine should not work against that plus variant. So again, we might expect new variants in the future, but as soon as that majority of the people are vaccinated, there is a chance that we can generate that herd immunity in order to avoid these new variants in the population. And with this, I would like to thank you all for your attention, and I will be happy to take any questions if you have.
Thank you, Dr. Aneesh. I have one question from Dr. Tiwari, Ramesh Tiwari. He wanted to know that whether for calculating the herd immunity, we consider only vaccinated person, or those who are, we could include those who are naturally infected.
No, for the herd immunity, it should be the ones which have been vaccinated with against COVID-19.
Mm-hmm.
So as I said, it's really important for the people which had the natural infection to get vaccination, because vaccinations are known to boost immune responses, and as the vaccine is not able to induce the neutralizing titers. The natural infection is not able to induce the neutralizing antibody titers as high as the vaccine. So it's important for those individuals which have been naturally infected to get the vaccine.
Okay. Second, being a viral disease, it is general perception that cell-mediated immunity is more effective. However, whatever the efficacy study and all we are doing, that is more based on the humoral immune response. So whether these antibody titer are correlate of protection, though you have discussed this, but still I want some.
Yeah, so far it's in the clinical trials and everything. Traditionally, it's very easy to take the blood sample and measure the antibody. So that has been done. And there are assays which have been established during the clinical trial sites, which are easy to measure the neutralizing antibody titers compared to measuring the T cells and following that. So that has been a traditional approach. And if you look at all the vaccines which are viral vaccines which have been approved for used in humans or animals, all of them induce the neutralizing antibody titers. So it's easy to, one of the reasons is that it's easy to measure the neutralizing antibody titers compared to T cells. Hopefully this will change in the future clinical trials. And the second reason is that the neutralizing antibody titers, when they were first tested, these vaccines were first tested in the preclinical studies in monkeys, they were found to strongly correlate with protection in large animal models. And that's why, and based on SARS-CoV-1 and MERS virus infection, it was found that neutralizing antibody titers have a promise, and that's why neutralizing antibody titers were always tested first, and they were found to be protective. So as you said, it's right that hopefully, maybe it's not enough. We need to go beyond neutralizing antibody titers, including T cells, as well as the other innate immune cells also.
Moreover, sir, one thing which attracts my thought that when we had swine flu in the last century, 1980s, I suppose, we have huge mortality. Somewhere I was reading that usually without any intervention, that disease was controlled within 3 or 4 year span of time. So whether do we consider such thing happening with the COVID also, that what the spikes we are getting or second wave, third wave, where it is going to end? Whether in the 2-3 years time frame, we could hope that, is there any predictive study made regarding this?
No, they have some modelings which has already been done, but and it's believed that it might become endemic like the flu. But one of the major differences between flu and this is that the flu changes so dramatically every year, antigenic drifts, so which is not the case with the SARS-CoV-2 virus. The only thing is this, say, occasional mutations. So that's why it's really important to get the vaccinations as soon as possible to achieve the herd immunity, so that we do not transmit the disease, and then that will lead to herd immunity, plus the new variants will not spread that fast, or they will not come up. So I believe in few years, if we have the global population vaccinated, hopefully this will transmission will spread, and then hopefully in a few years time it might become endemic, depending on how long the immunity lasts, and then we might have to get a booster dose maybe after 1 year or 2 years, something like that.
And sir, last question, one more question. Talking to you is quite interesting, and in that way I'm getting a lot of things from you. Sir, one thing I wanted to know, that suppose a vaccine has efficacy of 80%, 85%. Most of the vaccines are giving efficacy in that range.
Mm-hmm.
If the generation of immune response is a function of constitution of MHC of that particular subject.
Yeah.
So suppose if in simplest way we could say that if 100%, 100 people is got vaccinated with Covaccine, if the Covaccine has a efficacy of 85%, it means there is chance that 15% do not mount sufficient immune response, which could be protective. So keeping in mind the role of MHC in the immune response generation, can we resort to some other vaccine in the same subject? It's a technical question, but what I mean to say is that if somebody is not responded to a particular vaccine, is there any chance?
No, it's already happening actually. It's I don't know if you look at the data. So what they did in UK is it's mix and match strategy, in which they have mixed this Covishield vaccine with the messenger RNA vaccine, and they found that if you have the priming with the this Covaccine vaccine. This is the AstraZeneca vaccine, and then boosted with the messenger RNA. Yeah, these individuals, they use even better the productive antibody titers than neutralizing antibody titers than the original ones. And so this mix and match and boosters, they definitely they will happen, or it's being used in some countries actually already, especially in the US.
Thank you, sir. Thank you very much. If any question, anybody can ask.
I can see a question in the chat. So this is a question from Purushottam, I think. So why it has been seen that people got infected just after getting the vaccine. As I said earlier, that it's really important to consider the duration. So vaccines will not work immediately. It's not a magic wand that you will get the vaccine and it will work. You need to have at least the 2 doses of vaccine plus the 2 to 3 weeks more than after your second dose of vaccine in order the vaccines to fully generate the right kind of antibodies and T cells at a particular threshold. So for that particular duration, so 6 to 8 weeks time, since you get your first dose, until 2 to 3 weeks post the second dose is very critical, and you need to maintain the social distancing and all those things, wearing masks and everything at that time. And that's why what is happening, that people, if they are not maintaining that these precautions during that time, then they can contract that disease. So again, I would like to mention is that highlight again, vaccines do not, completely prevent the infection. You can still contract the disease, but they will prevent the transmission, they prevent the hospitalization, they prevent the symptomatic disease. But you can still get the infection. So this is how normally the vaccines work. You can still get the infection, but the vaccines, if they have generated the immunity, it will take care of the disease.
Thank you, sir. So if there is no more question, I would like to invite our Director Research, sir, for his closing remarks.
Thank you once again, Dr. Aneesh, the guest speaker for the day. I think you have already very well summarized how vaccines are being produced, what is the methodology, the further action, reaction within the body, clinical trial output, and how this is protecting our human being, and what the future looks like. Of course, that too has been projected in your talk. So very well summarized, and I think our faculty and our students have been benefited from this. And of course, I'd like to inform you that during these days, when our students as well as faculty were a lot much stressed, we also organized some de-stressing lectures, motivational talks for them. And of course, that help us in lightening their mood. And of course, I'm also taking first year classes. There too, the students are stressed a little bit. But now since the college is opening up, hopefully the things will improve, and they'll have the enjoyment of the campus life. Anyway, I once again thank Dr. Aneesh for sparing his valuable time for this talk, and our faculty colleagues, as and when have been assigned such a responsibility, they have delivered. Similarly, Dr. Pankaj over here, I assigned this responsibility to him. He has coordinated and done it, and we'll like to move ahead to have some more talks and panel discussion, as well as some virtual trainings, if we can organize for our faculty on some particular topics. Of course, since you are connected with microbiology, for microbiologists, we can have a short training, virtual, of course, which is possible these days. We shall be looking into it and sharing the information and talking to you later on. So sure. At the moment, I once again thank you, Dr. Aneesh. Thanks once again, and our coordinator for the today's program, Dr. Pankaj, our other faculty colleagues and students. Thank you very much. Thank you.
Thank you so much for your kind invitation, sir, and also organizing this nice, get together
okay.
today.
Okay, so Dr. Pankaj, we close the talk.
Yes, sir.
Okay, thank you very much. Thank you once again.
Thank you. Take care, sir. Thank you.
Okay,
Thank you, sir.
bye.
Bye.
Bye. Bye. Bye.